Hello, my name is Jihad, and this is my presentation for the histological and molecular analysis of early osseointegration dynamics at the Lewis rat model. So I was inspired to join the Bone Lab at UT Dallas after watching a Netflix documentary. It was talking about implant failures and how people suffer from that. So I was really curious to study why implants fail and how can we improve them. And this is what we do at the Bone Lab. So my work is concentrated on dental implants and their failure. As you can see in figure 1, so we have a dental implant, and due to many factors such as bacteria or the trauma after the surgery, we may have a failure that can lead to the peri-implantitis. This is like a late stage failure. We are targeting the early osseointegration, meaning we want to make that implant more susceptible to healing in a good way. So figure 2, I'm presenting the variations in dental implant models. We have different types of implants. We can use different animals. It could be mice, rats. We can use different dimensions of the implant themselves, different materials, and that results in different ways to present the data. For example, if we would like to do another research or to reproduce some results, the variation makes it really hard to predict the results of the study. So the goal or the objective of my work is to establish an oral osseointegration in the Lewis rat specifically to provide a reproducible baseline to track inflammatory response and healing process of titanium implants, meaning that we're establishing one type or one model so that its success can be reproduced and further studies can base their research on these findings. We're using obviously the Lewis rat, and my work was focused on the histological analysis and the molecular analysis. As you can see in figure 3, we have the histological analysis results where we can see that there is like new bone formation at 2, 7, 14, and 30 days. We can see the significant increase, and that was also traced back with the molecular analysis that confirms this because we study certain markers. Each marker, for example, can be associated with the pro-inflammatory stage. We have the macrophages stage, and we can also see that there are bone cells that were formed, and the results basically the evaluation of the Lewis rat model has demonstrated a 78.5% success rate versus 77.7 in mice model. The non-extraction model also is it reduces the potential of post-surgical complications. In a success rate more than 60% is considered similar to the human success rate. So this study basically it illustrated the dynamics of cellular and molecular events that are involved in early oral osseointegration. We were able to achieve high success rate of 78.5, meaning that we could prove that this model could be reused for future studies.